Good morning, everyone. My name is Gerhard Weiß. Today I'm acting as pro-rector of this university, representing the rector, Professor Pamela Habibovic, and I will chair this ceremony. I would like to welcome the candidate and wish you success. The supervisors as well, Professor Ilya Arts and Dr. Michiel Adriaans, and also, of course, the members of the degree committee. Dear guests in this room and online, family and friends, and colleagues of Balázs Erdős, welcome to Maastricht. The ceremony, for those who don't know it yet, the ceremony will start with a 15-minute presentation by the candidate, followed by 45 minutes questioning by the esteemed committee. After this, we, so the committee members, the supervisors and me, will retire for our deliberation and will then return to announce our verdict. Having said this, hereby I open this academic ceremony in which Balázs Erdős will defend the academic thesis Quantifying Metabolic Health from High-Dimensional Post-Meal Dynamics. May I invite you to present a summary of your study and the conclusions of your thesis in 15 minutes.
Dear Pro-Rector, dear members of the Corona, dear supervisors, dear colleagues, friends, and family, thank you for being here today for the defense of my thesis entitled Quantifying Metabolic Health from High-Dimensional Post-Meal Dynamics. My thesis revolves around the analysis of meal challenge test data, a complex type of experimental data that is more and more frequently generated in nutritional research in order to study metabolic health. In my research, I focused on developing analytical methodology to derive insight from such data in increasingly complex settings. First, let's briefly look at how meal challenge test data relates to metabolic health. Metabolism is how our body breaks down, stores, and uses the nutrients from the food that we eat. It is regulated by complex hormonal signals and substrate interactions. The ability to process food depends on many factors, such as age, sex, diet, weight, or even sleep. An important concept in metabolism is homeostasis. Homeostasis refers to the body's ability to maintain a stable internal environment in the face of external fluctuations, including changes in the availability of nutrients from food. If the capacity to maintain homeostasis weakens, metabolic health deteriorates, and correspondingly, the risk of developing metabolic disorders such as diabetes and other cardiovascular disease increases. Therefore, the capacity to maintain homeostasis can be seen as a measure of metabolic health. This concept is frequently used in nutritional and metabolic health research, for example, to study the glucose homeostasis. Glucose homeostasis is central to metabolism and metabolic health. On the slide, you can see a schematic of some of the processes involved in maintaining glucose homeostasis. In the post-meal state, that is the time period following the consumption of a meal, insulin, a key regulatory hormone, manages the glucose levels in the following way. After the meal traverses through the upper gastrointestinal tract, it appears in the gut. From here, glucose is transported to the bloodstream, and as the blood sugar levels rise, insulin is secreted from the pancreas and facilitates the uptake of glucose from circulation into tissues such as the muscle, liver, and adipose, where it's stored or used as energy. In the meantime, insulin also suppresses the release of stored glucose from the liver, since there is already an excess of sugar from the meal. Finally, as the glucose levels decrease towards the homeostatic level, insulin is also cleared from this system. If we take blood samples throughout this period and determine the glucose and insulin levels in these samples, we can draw the response of this system. This type of time series data shows us how well the body is able to regulate the glucose levels. In this way, the response profile can be used to distinguish the state of glucose homeostasis between groups or individuals. For example, the inability to return towards the homeostatic level, as seen in the green individual's case, may indicate that they are insulin resistant, a state of impaired insulin action which is associated with the high risk of developing diabetes and other cardiovascular disease. In order to enable such comparisons, the meal composition and other parameters of this experiment are standardized and controlled. For example, here a 75-gram glucose solution was consumed and we monitored the post-meal state for 2 hours. We call this test the oral glucose tolerance test, and it is frequently used to study the glucose homeostasis. Other meal challenge tests use more than just sugar, and in this case we may be interested in a comprehensive metabolomic response, not just that of glucose and insulin. Thereby we measure many more metabolites, such as amino acids or lipids. These standard meal challenges provide insight into the state of metabolic health. However, deriving quantitative information from such response profiles is challenging. For example, answering the question, how similar are the response profiles of the green and orange individuals, is already difficult to answer. This is due to the gap between the complexity of meal challenge test data and the corresponding analytical methodologies. Therefore, the analysis of this type of data has been limited to particular features of the response curve to be used in the comparison. A commonly used approach is to calculate the area under the curve, highlighted here with the shaded regions in the figure. This is indicative of the magnitude of the response. However, this approach fails to discriminate between different temporal profiles, even though the responses correspond to vastly different states of metabolic health. The property that we disregard here is the temporal dynamics, or in other words, the shape of the response curve. In addition, meal challenge tests perturb multiple metabolites simultaneously, such as glucose and insulin in this case. Therefore, considering only one of them while ignoring another may be misleading. For example, making a decision based on the insulin response in this case would lead to the wrong conclusion that the responses are similar. This property is the multivariate nature of the data. Finally, the importance of accurately quantifying such data is driven by a large heterogeneity that exists between individuals' responses. As this heterogeneity may reflect differences in metabolic health, it is important to be able to capture this. My research focused on addressing the methodological challenges related to these properties, the temporal dynamics, multivariate nature, and interindividual heterogeneity. In this way, my aim was to develop analytical methodologies to better characterize metabolic health from meal challenge test data. To do this, we proposed approaches based on mathematical and statistical modeling that was specifically designed to be able to handle the temporal dynamics, multivariate nature, and interindividual heterogeneity, the typical properties of such data. The resulting models can take data from a population of individuals as input and provide a simple quantitative summary of individuals' response profiles as an output. Mapping the input-output relation happens through a process called model calibration. During calibration, the model is fitted on measured data points of the meal challenge test. Here the black crosses correspond to the measurements and the continuous line to the model approximation. By doing this, we map the experimental data, including all its complex properties, into a simple summary where comparison becomes easy. This summary in turn leads to a more sensitive quantification of the response profiles compared to what was possible before. This way, we can now detect differences between response profiles that remained hidden with previously available analytical techniques. In the following slides, I will highlight some of the main results and findings in my thesis using our novel approaches. In Chapter 2, we studied the glucose homeostasis of a large group of individuals who underwent an oral glucose tolerance test. We used a physiology-based mathematical model to represent the processes involved in maintaining glucose homeostasis in the form of equations. We then developed a novel computational framework that enables the person-specific calibration of this model based on individuals' responses to the oral glucose tolerance test. The resulting personalized models, by accounting for the metabolite interactions and the temporal shape of the data, revealed a large heterogeneity between individuals' responses. In addition, the physiology-based nature of the model leads to direct insight into important processes of the glucose homeostasis, such as insulin sensitivity or beta cell function, in other words, the capacity to secrete insulin. Our framework to personalize such models is an important milestone towards providing detailed quantitative information about an individual instead of a population. In Chapter 3, we incorporated data from continuous glucose monitoring devices in the calibration of the personalized models that we developed in Chapter 2. These devices, worn on the arm or the abdomen, record glucose concentrations in the interstitial fluid every few minutes. We used experimental data where study participants underwent an oral glucose tolerance test while wearing a glucose monitor. In this way, we had two sets of glucose measurements, the conventional plasma samples and those from the CGM device. Therefore, we were able to compare the use of the two measurements in calibrating the personalized models. We found good agreement between the resulting insulin sensitivity and moderate agreement between beta cell function as measured by the personalized models. This is important because a conventional oral glucose tolerance test with venous sampling is quite invasive and labor-intensive, while the CGM device can deliver the time series of glucose data required for model calibration in a minimally invasive manner. This study was an important first step towards being able to derive detailed insight about glucose homeostasis through combining mathematical modeling with minimally invasive sensor technologies. The novel approaches that we developed using physiology-based mathematical modeling can provide insight from meal challenge test data, as we demonstrated in the first part of the thesis. However, the scope of the analysis here is limited to the handful of metabolites whose interactions we know about and therefore can encode in the form of equations. In the second part of the thesis, we focused on enabling a similar type of analysis of meal challenge test data, but now on the scale of state-of-the-art measurement techniques in metabolomics. These include hundreds of measurable metabolite species whose interactions we don't necessarily know. In order to do this, while taking into account the temporal dynamics, multivariate nature, and inter-individual heterogeneity as before, we developed a novel data-driven approach in Chapter 6. Here, instead of relying on our physiological understanding of the interaction between the metabolites, we make use of the large number of measurements to find the dominant patterns in the data. In this way, the novel technique can be used to analyze hundreds of metabolites measured in time series to identify individuals or groups with distinct metabolomic responses within a population. Our novel model also enables the quantification of differences between metabolomic responses of groups of interest. For example, in Chapter 7, we wanted to gain insight into the differences in post-meal metabolite responses between individuals with predominantly muscle versus predominantly liver insulin resistance. Our approach allowed us to test for differences between a 164-dimensional metabolite response to a meal challenge test. We found distinct metabolite profiles that could not be identified using previously available analytical techniques. These findings help us understand the physiological differences underlying these different states of metabolic health. This model can now scale up this type of analysis of meal challenge test data to the hundreds of measured metabolites after various different meal challenge tests and experimental settings. To conclude, in this thesis, we contributed multiple novel methodologies in the form of mathematical models for the analysis of meal challenge test data. We demonstrated that our methods, by accounting for the complex properties of meal challenge test data, can lead to insight about metabolic health. Our models and findings have potential for advancing research in nutritional and metabolic health, as well as to enhance personalized nutrition technologies that are designed to optimize health through diet. With that, I thank you for your attention, and I give back the word to the prorector.
Thank you for your presentation. The opposition will be opened by Professor Dr. Ralf Peters, Professor for Mathematics and Knowledge Engineering from the Department of Advanced Computing Sciences, Maastricht University. Please.
Thank you, Mr. Prorector. And let me first start with congratulating you with a very nice thesis. And I think, well, if you see the volume of this, I would almost call it an obese thesis, which fits the topic pretty well. Also, my congratulations to the supervisory team. I think it's a very nice collection of work, and I've at least enjoyed reading this a lot. I would like to question you and discuss with you some of the topics in Chapter 8, and there you have a number of models that you start to compare. And I think the main purpose of that chapter is to see if you can actually get to hybrid models where you mix two different modeling approaches. One was bottom-up, the mechanistic type of models, and the other one top-down, data-driven models, and then see if you can use different things to bring them together. And first of all, I think it didn't work very well, because whatever came out was either on one side or on the other side, you know, depending on the type of model you have. You have two different hybrid models. But suppose it did work well. Then I was wondering about the model usage. Suppose you got a model out that was hybrid and that had the mechanism inside, the differential equations, but also on top of it used the other features for slight error corrections. What would you do with the model eventually? What is the main use of it?
Dear highly esteemed opponent, thank you for your kind words and your question. That is indeed the case. We haven't found a great improvement in this application for applying the data-driven methodology on top of the mechanistic model. However, the main goal of this predominantly is to find weaknesses in the underlying mechanistic model where its predictive power is limited. So settings such as free living conditions, as opposed to the simple oral glucose tolerance test that our model we found can explain it very well in itself, the mechanistic model. In free living conditions with different meal compositions, meal quantities, proximity to sleep, exercise, etc., we might find that this simple limited mechanistic model isn't enough to sufficiently explain the response. In these cases, what we would expect is that the data-driven model would find the features that are missing from the mechanistic model, so to speak. We're not encoding them in the equations. That helps us explain the missing part of variability. For example, if a larger meal is consumed, and let's say our model cannot account for this, we would find meal content, or quantity, as an important predictor. For how towards the use of this model, I think first and foremost, I was interested in finding the limitations of our mechanistic model. So it could be used for building better models, what's missing, and so on and so forth. From the other perspective, if it works well, if it's quite predictive, we could then package it and just use it as a predictor. And once we measure these features, I assume these are, I imagine that these features, the extra features from the data-driven model, are related to everyday life, usually easy to acquire. We could use an already built mechanistic model with the trained on OGTT once, and then tune its prediction based on real-time data from these other features that are easy to acquire, such as when the meal was consumed, how big the meal was, etc.
Okay, so I get that if you were to use elements, features of, let's say, the metabolites that you mentioned for the other chapters. But here in this chapter, you use these generic kind of things like age and body mass index and a number of those kind of things. So how would that link to a mechanism? Because that's what you gave as a motivation.
Yeah, so in terms of relating to mechanisms, for example, in our mechanistic model, we do not account for differences in, let's say, weight or BMI. So let's say our model, our data-driven model seems to favor, which it actually also does a little bit, although the predictive power is limited. Seems like BMI is an important predictor. That may highlight that there is a mechanism that kind of modifies glucose control, glucose and insulin interactions, depending on BMI. And this is completely feasible, and it is actually true. And we could find something like this if we find that this feature pops up in the data-driven model.
Okay, I understand it. And then if you get a better prediction of the curve that you actually observed and which you measured, so you have those data, then what? We have a better prediction. So yeah. Are you going to use the predictions for something, or are you going to use something else?
Yes, we can. So the main idea behind these personalized predictors, so the ones that focus on prediction, is to be eventually used in technologies related to personalized nutrition, for example, so that a participant enrolls, the model is running in an app, let's say, or on cloud. They underwent an oral glucose tolerance test, so we have the mechanistic model calibrated, and then one day to the next they put in their diaries and these informations, their weight and whatnot, and then we can give accurate prediction what the response would be if they ate something, and so we can kind of modulate this important target of how the glucose and insulin responses will be for a hypothetical meal. And so we could reduce the glucose response to something that's healthier.
Okay, I see that. Then I would like to switch topic a little bit. I think there's a little bit of time for that left. And my next question is going to be on Proposition 6. So I would like one of the paronyms maybe to read out Proposition 6.
Many different models may describe the system of interest. Consequently, model selection should prioritize model reusability and interoperability.
Yeah, thank you. So it's about prioritizing this reusability and interoperability when you select a model. And I had actually two questions. So prioritize over what? And the other one would be, can you also indicate how that plays out for the models in Chapter 8, concretely?
So regarding the first part of the question, prioritized over what, I think of this in the form of model selection, for example. But there's also another component related to the actual software and how it's packaged and shared with others. But regarding model selection, this is a very difficult topic of trying to find a model that fits and explains well, that you can then reuse in different settings. So, for example, if your model is too complex, it might explain your data and your processes well. But if then in another setting you have a little bit more limited data, you will not be able to use the same model for that purpose. And so here, choosing the more limited model would be favoring or prioritizing reusability. So a more parsimonious model, although this is quite generic answer for a model selection, but it's a very important topic in this setting because models can go really complex. Regarding the software, it would be very important that everything is reproducible and reusable irrespective of the software that's being used or the data that's being used, and that is also a very difficult topic and question, but that's incredibly important so that these models will ever see any use outside of the same lab.
Okay, thank you very much for your answers. I'm satisfied, and I return the word to the pro-rector.
Thank you for your questions, Professor Peters. And by the way, also thank you for chairing the assessment committee. The opposition will be continued by Professor Dr. Martijn Brouwers, Professor for Internal Medicine from the School for Cardiovascular Diseases, Faculty Health, Medicine and Life Sciences from the Maastricht University Medical Center.
Thank you, Chair. So, dear candidate, I also would like to congratulate you on this very nice thesis. I read it with great interest. I also would like to congratulate your team on this really great result. I would like to start off with a more general question on your thesis. I think you quite convinced me that using these temporal dynamics after a meal and also take into account these multiple metabolites are really helpful to detect inter-individual variability. But what about intra-individual variability? What we, for example, know in oral glucose tolerance test, if you repeat that within one individual, well, the responses in glucose, they differ quite a lot within one individual. So how does that affect your models? That's my first question. My second question would be how to deal with that.
Your highly esteemed opponent, thank you for your kind words and your question. Indeed, the intra-individual variability we did not discuss as much in the thesis, and there are practical reasons for that. And I completely agree, it's at least as important as the inter-individual variability. So there are kind of two aspects that I would like to highlight. One of them is that when we talk about intra-individual variability, in a single set of response, we would look at the curve, the shape of the curve, the different measurements in the same test. So that we can completely account for because that's the main goal of the temporal dynamics, the models that can quantify that. But perhaps the more interesting aspect is how repeatable the test itself is. And we haven't tested that because we didn't have access to data with repeated meal challenge tests in the same condition. And I think that is the single biggest limitation towards the usability of these models because we cannot quantify how much of this variability relates to the mechanisms that we're interested in, let's say glucose control, compared to other day-to-day variation, which is still very important, like stress or sleep or whatnot, but we cannot kind of separate the sources of variability because we only have a single set of response. I think the next step in any such analysis would be to thoroughly evaluate this if we have data where participants undergo the same meal challenge test with no intervention in between. So that would be extremely interesting to look at.
Yeah. Of course, you had to deal with the data that is present in these cohorts that you used. So one cohort you had the OGTT and another one what's the mixed meal. What would be your ideal meal test? Is there an ideal meal test for you to use?
I think from my limited understanding of the physiology, I think the more realistic, although it depends on the aim. So of course, if we want to study glucose homeostasis, maybe the OGTT is ideal. But I think for generalizing towards nutrition and nutritional advice, something like a mixed meal challenge test would be ideal. That also contains protein and lipids. And there's a lot of literature out there on the ideal composition of these challenges. But of course, in the studies that I used, they were specifically designed to study particular properties in the responses. So, for example, OGTT for glucose homeostasis or the type of meal challenge tests in chapter 7 were, for example, designed particularly for the type of insulin resistance. They were already hypothesized to have beneficial effect on one or the other groups.
Well, that's a really nice bridge, actually, chapter 7. In that chapter, you made a distinction between two types of insulin resistance, so the muscle and the liver insulin resistance. And this distinction was based on the OGTT. And I'm always a little bit fascinated by that because it appears that from the OGTT, you can derive all kinds of indices. So the HOMA, the Matsuda, but also the HIRI and the MISI. How valid are these HIRI and MISI for their purpose?
That is a very good question, and probably I'm not the best person to answer it, but in my opinion, with the current evidence, including that of the groups that I've collaborated with throughout my PhD, is that these are indeed distinct phenotypes now that are defined based on these tertiles of MESI and HIRI. So we do see distinct differences repeated across different study populations, both in their fasting conditions previously, not replicated in our chapter 7, and in their postprandial responses. So they do seem to identify these MESI and HIRI-based liver and muscle insulin resistance groups, kind of distinctions between them. It looks like the liver,
Well, that appears a little bit like a post hoc conclusion that based on the data that you are getting, it appears to be good. But if you look at the original paper, and that's what I did, where they more or less developed this concept of this HIRI and MESI, what they did is in groups where they performed this hypoglycemic, hyperinsulinic clamps, really which is the gold standard to quantify hepatic and muscle insulin resistance. And there they used these indices and correlated them with the golden standard. But for example, if you look at HIRI, the correlation with liver insulin resistance is 0.64. Wow, okay, I would say. But for the same HIRI, the correlation with muscle insulin resistance is 0.55. Well, it's a little bit less, but still, I would say it's the same. So yes, I do agree with you that in your post hoc conclusion, there appears to be differences between the groups. But if you purely look at the correlations, I would say the HIRI reflects both liver insulin resistance and muscle insulin resistance. What do you think?
Generally, I agree. And what we see is that also there is, while these associations replicate to some extent, there is variation between study to study, how high this correlation is. But I think we also have to remember that this is purely based on an OGTT, which includes many other factors compared to the clamp, which is less physiological. So the OGTT contains properties of the gastrointestinal, upper gastrointestinal tract, such as meal emptying, which seems to have a big gastric emptying, seems to have a big effect, for example, on the responses. So it's quite some physiology in between going from one to the other. So how high that correlation is or could be, or how high we should expect, is a little bit, I think, debatable. But I think from the evidence still, I think that these are quite distinct groups, at least based on how they are defined. Whether or not this is going to be, so we also see, of course, the intervention. We did an intervention as related to the data in chapter 7. And they did seem to also respond differently. So these muscle and liver insulin resistance groups were defined based on an OGTT at a baseline of a trial. They were prescribed diets for 12 weeks, I believe, which were hypothesized to be better for one or the other. And they do seem to respond differently. So there seems to be a benefit for them to take a more personalized diet based on their phenotype. So there is plenty of evidence, I think, that suggests that this makes sense. But indeed, how they are defined could benefit from some quantification of the uncertainty, let's say.
Okay. Thank you very much. I'm very pleased with all the answers that you were giving me. I give the word back to the pro-rector.
Thank you, Professor Brouwers. The opposition will be continued by today's second online candidate, Dr. Susan Wopperijs. She is principal scientist in systems biology at TNO, so the Netherlands Organization for Applied Scientific Research. Your questions, please.
Thank you. Dear candidates, first I would like to congratulate you with your very nice thesis and also your team. I'm very impressed with the contents of your diverse modeling approaches that you applied on high-dimensional post-meal dynamics. I have been working with challenge test and post-meal dynamics for almost 15 years now, and I have been searching for a long time for data analysis approaches that could get more out of my data. And I think this thesis is really a gold mine with multiple very interesting solutions. So thank you for that. For my questions, I would like to discuss with you on chapter 2 and 3, and maybe a little bit on chapter 4. I was actually quite triggered by a statement that you made, or at least it was summarized under the author summary page. Author summary, which is put on page 31, the first line. And I will state your statement, which is, furthermore, the models can be quantified from comparatively non-invasive sampling techniques and may be readily transferable to the clinic. And I'm very interested in what your opinion is, what is needed to have it transferred to the clinic. So therefore, I have identified several sub-themes that I would like to discuss with you. And the first is about to get those physiology-based mathematical models, the personalized EDAS models, transferred to the clinic. So do we need, I'm interested, do we need an oral glucose tolerance test for that? Do we need maybe a mixed meal challenge test? Or can we do without a challenge test? Or what is your opinion on that?
Dear esteemed opponent, thank you for your kind words and your questions. I would relate back in terms of the translatability to clinic of these models. I would refer back to the previous discussion. I wrote they may readily transfer to the clinic, primarily because of this quantification of the uncertainty, the replicability of the test, the challenge test itself, the OGTT, is not very well defined or evaluated. So there has been many studies that looked at the reproducibility of an OGTT under the same conditions, but it matters a lot what analytical technique is used to check the reproducibility. So for example, more frequently they look at whether the diabetes status based on WHO or ADA guidelines, they hold after they repeat the test. But this is of course extremely crude compared to the sensitivity that we can provide with these models. So before we can say anything about its use in clinical or any other setting for that matter, in my opinion, we should really quantify how sensitive these approaches should be. So we can quantify very nuanced differences in temporal shape, but do we want to quantify that? Does that relate to the actual mechanism that we want to target with an intervention or use in diagnosis, or is it just some day-to-day variation that we shouldn't care about? And this is impeding any translation to the clinic currently, I think. Regarding what is... Yeah, sorry.
Sorry. Go ahead.
What is needed, what kind of data or challenge test is needed, I do think that something like a 5-point standard oral glucose tolerance test is worth to conduct in a clinical setting because it gives us a lot more insight compared to, let's say, fasting values. And of course, there is no or very little benefit to applying the model. We could apply the model on just fasting data, but then it would predict the same kind of response, just at a different amplitude for different individuals, and it wouldn't be very diagnostically useful. So I think a challenge test is still ideal, but there's of course now more and more sensor data becoming available because there's debatable how uninvasive or less invasive these challenge tests are. I think they're quite invasive. They're much less invasive than the clamp, but they are still quite invasive. So doing anything towards more minimally invasive sensor technologies to enable the calibration of this model, which is what we're trying to do in Chapter 3, would be beneficial.
Okay, so yeah, that would be my next question. So, okay, you prefer the 5 points oral glucose tolerance test combined with continuous glucose monitoring data. What other measurements are essential to that? So is it only the CGM, and does it matter what type of sensor? Yeah.
So for now, I think the benefit of applying the models that we have, and this has been a critical point of our Chapter 3, is that we need the insulin measurement. So, and this is more a biologically or physiologically inspired choice, because in particularly in the populations that we looked at, kind of individuals with obesity or insulin resistance before overt clinical disease or type 2 diabetes, it is incredibly important to be able to distinguish between the state of beta cell adaptation, I think. So we could see the same, very similar glucose responses with widely different insulin levels. And so for me, I would prefer to have the insulin levels always to calibrate these models because it can capture much more important differences. The CGM data, CGM glucose, instead of plasma glucose, I think is already feasible to replace it. Of course, in our chapter 3, this looks a little bit silly because we already have the same plasma insulin measurements, so we need the test anyway. But our hope is that eventually insulin will also be able to be measured in like minimally invasive sensor technologies. I think there's experiments on dried blood spot, C-peptide that could be used as a proxy, and so on and so forth.
Yes, I do agree. And can you use any CGM device, or should you also adapt your model to what type of CGM device?
I think that is a very important question. I cannot directly answer it now because I had limited access to the different devices. But from the literature, there seems to be large variability between manufacturers, between where the sensor is worn, but there's conflicting information. Some groups report that they find good agreements between even different devices, different manufacturers. Some other groups report widely different responses with the different manufacturers or devices worn on different body parts. But this needs to be tested, I think, for the particular application. In general, to put them into the model, this is readily available. You might need to change a little bit the diffusion from plasma to interstitium compartment that we have in the model to accurately describe a particular type of device.
Yeah, so and then I'm wondering, having your model, your personalized EDAS model, I was wondering, what would you bring to the patient? So what would be the output of your model? Is that the different outputs, the K1, K5, K6, or K8? Or would that be maybe the liver insulin resistance versus the muscle insulin sensitivity? Are there subtypes, or how would you provide the information back to the patient?
Dear candidate, may I ask you for a short
for now, I think we can only provide these parameters that we estimated, insulin sensitivity and beta cell function, maybe gastric emptying, although that needs to be validated. In order to go down to the level of liver muscle, I think we need to improve the endogenous glucose production part of the model. So for now, I can only say that this would be useful information, maybe not to the patient itself, but their clinician.
Thank you very much for your answers. I will give back the words to the prorector.
Thank you, Dr. Oppereis. The opposition will be continued by Dr. Pietro Bonizzi, Associate Professor at the Department of Advanced Computing Sciences, Faculty of Science and Engineering from this university.
Thank you, Mr. Prorector. Dear candidate, dear Balázs, also congratulations from my side first for your very nice work and very nice manuscript. I really enjoyed to read it. Also congratulations to the supervisory team from my side. I did really enjoy the focus on the, let's say, engineering, focus on the data science, but also your real effort in trying to combine those two fields. It's very, very nice piece of work. I have one question more about the engineering and the mathematical models. In some parts of your work, you perform sensitivity analysis of your models, and my impression is that you focus on changing one parameter at times, which is very common practice in engineering. At the same time, when it comes to the human body, it's a very complex system. So you may wonder what is the effect of interactions between different terms. So what is the possible presence of confounders, which in statistics means, okay, maybe some factors appear to happen important where actually they're not. It's simply because of the effect of another term, and some factors may actually be hidden by the effect of other terms. So what are your speculation on possible biases eventually of analyzing one parameter at a time on your results?
Dear esteemed opponent, thank you for your kind words and your question. Indeed, we chose to do only local sensitivity analysis in our personalization framework. So the goal there is to reduce the number of parameters that we can reliably estimate in an individual setting, so limited data setting. The reason why we only did the local sensitivity analysis instead of the multi-parameter sensitivity analysis or global sensitivity analysis is because these models themselves have been developed prior to our work, and they already conducted all of this. And we were specifically focusing on aspects that have an impact on the personalization. But to give you some idea about the interactions, there are some parameters that show sensitivity when they're co-estimated with the different parameter. So that is happening. But we only estimate such limited number of parameters and choices of parameters where this should not have a high effect on the outcomes. But for example, we do estimate 2 parameters pertaining to insulin sensitivity, insulin secretion, and those do seem to correlate. So this is actually something that we've kind of fixed from chapter 2 to chapter 3 in order to improve this. We only estimate one of them. Nevertheless, the results didn't seem to change much. We use as validation other measures of insulin secretion that are already used in research and clinical settings. And we associate the parameters with those other measurements or other metrics. And we find similar associations between all of our personalized models from chapter 2, 3, 8. So they seem quite similar. So it seems quite robust.
Okay, thanks. My second question is more about the data science part, especially about chapter 6. And in equation 6.1, you present a mixed model. And my impression is that you treat every variable in your model both as a fixed term and as a random term, right? Fixed and random effect, which is strange to me because one variable should be either one or the other. So why did you, is my interpretation correct, first of all, and why did you choose for this way?
Yes, it is absolutely correct. And it is quite unusual to do this in the statistical literature. The reason why we do this, so this relates to this concept of linear mixed models are frequently used as random intercept, random slope models, for example. So the motivation behind this is that, for example, a random intercept is we can account for differences at baseline of an individual, but their response profile would be the same. If you go to random slope model, then you would have differences, you can quantify differences in baseline, so the fasting value, the first time point, but also differences in the shape. And this is why we incorporate the random effects. They allow us to capture person-specific profiles. And why it looks strange is because we use these generic polynomials as splines, which have 2 or 3 coefficients, depending on the complexity of the model, so linear, quadratic, and cubic maybe. And for all of those, we have to include them as random effect in order to enable that the individuals show a different profile compared to the fixed effect, which would be the kind of population average.
Okay, but for example, if I look at time, my impression that you consider time both as a fixed effect and as a random effect, while to me it should be either one or the other. So because if you sample at a regular time for every subject, then time is fixed. While if your sampling time is different for different subjects, then it's okay. You should consider it as a random effect. But you should consider both at the same time.
So regarding the sampling regime, with these mixed models, irrespective of fixed or random effect, we can use any continuous time measured at irregular samples because time is treated as a continuum instead of time as factor. But regarding to why we do this why we also keep time as a random effect is again going back to what this allows us, so the time effect is basically the shape. So
no, but is time fixed for every subject or not?
No, so
it's a random
time
effect.
is also a fixed and also a random effect. And what that enables us to do is if we have a population average profile like this, an individual can deviate from that. And encoding time as also a random effect enables this. This is basically a random slope model where the slope is a little bit more complicated than just the linear.
No, I understand, but the way the way you acquire your data is such that every subject is sampled at exactly the same time point.
Yes.
So time should be fixed because you don't have information about the randomness of time.
This randomness does not correspond to the randomness in sampling regime. The only thing it enables is for us to quantify a coefficient specific for the individual, whether their response is, let's say, their rate of change is higher or lower. It has no relation to the sampling regime or like when it was sampled.
Okay. Yeah. Okay. Thank you. I give the word back to the product. Thank you.
Thank you, Dr. Bonizzi. The opposition will be concluded by our last opponent for today, Dr. Kiki van der Heijden, assistant professor at the Dunders Institute for Brain Cognition and Behavior, Radboud University, Nijmegen.
Thank you. Dear candidate, dear Balázs, also I would like to extend my congratulations to you and your supervisory team for your thesis, which stands out not only in terms of quality of the research, but also in terms of quantity of the research, indeed, as was acknowledged before. In terms of my questions, I would like to go back to chapter 8, which is where we started, I think, with the opposition. And as was discussed already, here you start to combine the mechanistic approaches with data-driven approaches, as well as hybrid approaches. And I wanted to ask you about your selection of the data-driven approach. So you analyze the data with gradient boosting regression, but your mechanistic approach is specifically focused also on capturing the temporal dependencies. And throughout your thesis, you are stressing the importance of including these temporal patterns in the analysis. And therefore, I was wondering what your motivation was for choosing this particular approach, and looking back at your results, whether you could elaborate on alternative approaches which are data-driven, but which could capture these temporal dependencies.
Dear esteemed opponent, thank you for your kind words and your question. First off, I would say the main motivation for choosing gradient-boosted regression is for its interpretability and the explainability of the predictions. So like it provides us very easy to understand feature importances, which was the key outcome that we were looking for. Nevertheless, I completely agree. It goes against with what I'm trying to advocate for, as in the temporal dynamics, because this model predicts the residual per time point independently. So it does not hold any relation to the temporal dependence whatsoever. To combat this, we did try other ways of combining the residuals into a target that reserves temporal dimension. So instead of predicting per time point, we were trying things like taking the slope between predictions, thereby linking the targets and their temporal dependence, and we did not see much improvements. So more or less what this chapter got reduced to is just a showcase of how well our personalized mechanistic models work, but this is also something that we did not expect, and that's why there's not much to be explained. What we did try and did not end up in the thesis is deliberately perturbing the personalized models. So we know we quantify a good, reliable measure of insulin sensitivity, and we deliberately change it to something wrong, and then try to recover that with the gradient-boosted regression, recover its prediction, and hope that the feature that comes up as important or predictive relates to insulin sensitivity, which we disrupted. And this was the case in simple simulation studies, but we could not show this in the simple OGTT setting that we tried there. In terms of other models, There are, of course, a plethora of different other more complex, more flexible models that can directly account for the temporal dynamics. Deep learning is one of them that everyone is trying now because there's quite good training schemes now, including data augmentation schemes where you can train in a data-limited scenario, which we have. But we opted not to try any of this because of the limited scope, because we only had the OGTT. If we had free living conditions where we expect more differences, we would have gone and tested some benchmarks against different models to see what the effect of accounting for the temporal dynamics in that step as well would be.
Okay, thank you. And then for the same chapter, besides the choice of data-driven approach, you mentioned also in the discussion that part of the inability of explaining any additional variance with either the hybrid approach or the data-driven approach may be that the features that you included in the data-driven approach may be falling within the same causal pathway as the mechanistic approach. And I was wondering whether you actually looked into that, and if not, how you would approach that.
Yes, that is a very important thing we did not do. The reason why we hypothesized that they fall into the same kind of causal pathways is that, for example, the limited predictive, the features that had a limited predictive power, everything had a limited predictive power, but what we found were things like diabetes medication popped up as an important feature to explain the response, and that of course makes complete sense because we're trying to capture the glucose regulation. So you would expect worse glucose regulation of someone who undergoes diabetic medication treatment. And so things like this we could directly show, but we did not follow up further because I think the setting here is too limited and there is not much gain to be using the data-driven model whatsoever. This would be interesting to do in another scenario where we have variability that we already know cannot be explained by the model. So things like looking at mixed meals with a model that explains only glucose and insulin, glucose regulation, or going to a complex real-life setting where the meal composition quantity can differ, maybe the person exercised before, and so on and so forth. And there, the same type of features that we had here might have much more relevance because they capture their everyday life. So for example, things like their habitual physical activity, which we had access to, would be an important predictor there, I think, I would hypothesize, but not for such a controlled standard clinical test where we may not expect to see this as much, or it's already captured by this kind of similar causal pathways in the model.
Okay, thank you. Then maybe I would like to ask you a final question about proposition 8. If the parent could read the proposition.
The integration of minimally invasive sensor-based measurements with mathematical models is key to enable translation of findings derived from invasive experiments in the research setting into practical products and treatment approaches.
Thank you. So here you are describing the translation from research to clinical or practical applications. And in your thesis, you also stress several times that nowadays there is a lot more data available thanks to these new sensor technologies and also much richer data. And you are linking it here specifically to mathematical models. And as you know, my background is especially in machine learning and artificial intelligence approaches. So I wanted to ask you how you see the role of machine learning or AI in addition to mathematical models, instead of mathematical models, for making this transition from research to application.
I think that is going to be a combination of mathematical modeling, mechanistic modeling in particular, where we have some underlying physiological understanding, such as the ones we use, in combination with with machine learning, deep learning, is an extremely prevalent field of research now. In particular, I would highlight, for example, the use of physics-informed neural networks, for example, and related aspects of modeling, where you can get the best of both worlds. You can keep what you understand, and you can build on that with the predictive model.
So, Balázs Erdős, the time appointed for... Oh, thank you. Balázs Erdős, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room. Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. To the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
From tomorrow, you can decide. That's nice.
didn't know that was so old.
And they had to post it also, so they sent it to Norway. No wonder it was so good. But it was still a comedy.
Yeah.
No wonder it was so much.
I actually thought they were really big. And I didn't need to say,
Oh, customs.
yeah, customs. You can't really get a lot of stuff for the guy who's doing something. Now it's a bit I need a tablet for my bike for 6 months. Because I could only get it from
Also, you should stay in
Lagos.
Yeah, they are
Now it's in customs.
Oh, yeah, also Finnish, yeah.
That's because he already worked for the UN.
Oh yeah, the weather is amazing. It's not.
No, yes, it was a second contract, so if they wanted to extend it, then it has to be.
So I could be excited.
So they give him 4 years and he can leave then. But if they only give him 2 years, then he's going to extend. Specific bills.
People are also huge there, so I can't get the.
Oh, yeah. It's okay.
Please all rise. Please be seated.
So, Balázs Erdős, the degree committee here present has assessed the quality of your thesis and your defense in view of its positive verdict and taking into account your previous qualifications. The degree committee has decided to grant you the degree of doctor. Professor Arts is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Balázs Erdős, the degree of doctor and grant you all rights attached by custom and law.
Thank you.
As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Please be seated.
Dear Dr. Balázs Erdős, dear Balázs, I have the honor to be the first to congratulate you with this great achievement. A beautiful and rich thesis, both in terms of volume, diversity, and quality, and an excellent defense. I was expecting nothing else from you, but still, you did it. Congratulations also to your wife, Petra. Great to see you here. And other family and friends all over the world. Also on behalf of my co-promoter, Dr. Michiel Adriaans, I would like to sincerely thank the members of the assessment committee. Professor Ralf Peters, who was also the chair of the committee, Professor Maria Suarez-Diaz, who could unfortunately not be here today, Professor Martijn Brouwers and Dr. Suzanne Wopperijs, here present online, Dr. Pietro Bonizzi, and Dr. Kiki van der Heijden as additional member of the Defense Committee. Thank you all so much for the time you spent on this and for contributing to this great defense today. Dear Balázs, your adventure in Maastricht started almost 6 years ago. We had a vacancy for a PhD candidate position, and there was a huge pile of applicants, but there was really only one suitable candidate who met the criteria and the characteristics that we were looking for. And that's a bit tricky, of course, if there's only one suitable applicant, so we were a bit worried. But after having spoken to you, there was no doubt. We didn't need to look further. We had found our one needle in this huge haystack. You started to work on the Top Institute Food of Nutrition funded person study program, led by Professor Ellen Black from the Department of Human Biology. The person study was a personalized dietary intervention study, you mentioned it today, with a large consortium of researchers involved, and you were basically hired to support and do the analysis of all the complex data that were being generated with such a study. I mentioned that you started 6 years ago. And if people think that 6 years is a pretty long time to complete a PhD, in this case, they are wrong. You not only developed novel data analysis and modeling approaches for the person study, you also cleaned data, CGM, and helped others to apply your methods. And in the meantime, we actually extended your position twice because we wanted you to work on 2 other projects. The NWO-funded Matroska project, led by Professor Natal van Riel from TU Eindhoven, who's in the audience here today, and the DTL-funded hotel project with Professor Aage Smilde from the University of Amsterdam. So all in all, you wrote 12 excellent papers that are almost all published now. So if you think this thesis is heavy, it doesn't even contain all the work that you actually did during these 6 years. The reason we asked you to stay on for these other projects was, of course, that we were extremely impressed by the way you worked. You were highly independent in formulating your research questions and finding and testing novel approaches to address these. You're able to cover not only the technical details of the modeling, you also understand the experimental methods that are used to collect the data, and you have a really large degree of biological understanding of the system that you're working on. And that was also mentioned today, that is really rare to find that in one person. So not only you are an exceptional scientist, you're also extremely helpful and a great collaborator. You formed a great person-study tandem with Bart van Sloen, who is here as your paranymph, and who defended his thesis last year. And you contributed a lot to the development of the STEM graduate school of the Faculty of Science and Engineering that started in September 2020 in the role of founding PhD representative. Balázs, you are always very serious and critical, and I was sometimes worried that you did not enjoy this PhD period to the fullest. And yes, you are very serious and critical and perhaps even a bit obsessed, but luckily not only with work. You also do other stuff outside work. I think you're a bit obsessed with bikes. Who else has a picture of a bike or their bike as a background on their laptop screen? And most of all, you're a little bit obsessed with coffee. You're the leader of the daily MaxBio exodus to Banditos, apparently the only place with drinkable coffee around. At least if we have to believe you. And you always make sure that wherever you go in the world, you know a good coffee place to go to. So you are serious and perhaps a bit quiet. And then all of a sudden, last year at the FSE party, there you were. I'm not going to say anything about what happened, but I think everybody had a fun evening. So Balázs, it has been a great pleasure to work with you over the past years. You're an excellent scientist, I already mentioned it. And I hope we have also convinced you of that fact. Believe me, it was not always easy to convince you of that fact. Unfortunately, in the end, it was the collaboration with Age that pointed you to interesting research opportunities in Norway and away from MaxBio. But I do think that was a great choice that I fully supported, as long as you promise at some point to come back to Maastricht. So I hope you have enjoyed this day so far. I hope you will have a great reminder of the day. You can be extremely proud of what you have achieved. And I wish you all the best in your further work and life.
Thank you very much.
Thank you for this laudation, Professor Arts. So, dear Dr. Erdős, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. I also would like to extend my congratulations to the supervisory team and, of course, to your wife. Last but not least, I would like to give the two online opponents the opportunity to congratulate you. Maybe we start with Dr. Woppereis.
Honored that I am one of the first to congratulate you. I would like to congratulate you, Balázs, and also your supervisory team, of course, and your wife, with this perfect defense, I would say, and a very nice thesis. And good luck with your postdoc position in Oslo, what I understood.
Thank you very much, Dr.
Thanks, Professor Brouwers.
Yeah, so Balázs, congratulations with this very great efforts. So I was really impressed by your defense and also by your thesis, of course. You did a really great job, and what I particularly liked is that you not only know a lot about all the models that you used and constructed, but you also know quite a lot about the methods and the biology behind it, and that's quite unique, I would say. So keep that, because that's really important to be able to communicate with other types of researchers and also to translate your findings to clinical practice and society. So keep on. And enjoy today, of course.
Okay. Thank you. So herewith I close this ceremony. This was the official part. The committee and you will now take a picture. First in this room, together with the two online opponents. Then we take a picture outside at the staircases. The audience, in the meantime, can already go to the rafter where the reception takes place, and where we will then show up after